Welcome back to the molpigs podcast. Today our guest is Damien Woods. Also with me today are Boya.
Hi.
Georgios.
Hello.
And I'm Will. Damien is based at the Hamilton Institute at Maynooth University, Ireland, where his group, Tap Dance, conducts research on molecular computers, collections of carefully engineered DNA molecules that bump into each other and interact in a test tube to solve some computational problem. His focus is on both the underlying computational theory and implementation in the lab. Previously, he held positions at Inria, Caltech, the University of Seville, and University College Cork. Damien, hi.
Hey, it's great to be here.
So let's start with an important question. Where did the name of your group, Tap Dance, come from?
So that happened when I was at Inria in France. And Inria have a tradition where where groups have names that are not the names of the PI, just, some acronym-based names, because computer scientists just love acronyms.
So what does it stand for?
It's the theory and practice of DNA computing engines. So we do both theory and practice, all right? So we do mathematical style theory, proving theorems for models of computation for DNA and molecular computers. And we do the practice of that. So we try to build those in the lab. And then the final part, so theory and practice of DNA computing engines, is because generally there could be a fuel that pushes these things forward.
You've mentioned a bit, but could you tell us a bit more about your group's research and what you're doing?
Yeah, so we're mainly driven by doing computer science theory for models of computation. Some of those are molecular computing inspired models. Some are not. Some are more traditional models like Turing machines or cellular automata. And we also do experiments. So we try to build nanostructures in the lab, build DNA computers in the lab, molecular robots, self-replicators, things like that.
Damien, you've been at many institutes according to your bio. How has that changed your research interest over time, being in so many different environments and research groups?
Okay, so I guess, so that's a tricky question. So definitely some of the institutes influenced my research directions, but not all. So I guess if you look at my publications, you'll see a diversity of stuff there perhaps. But for me, it's all following a common thread that has been there right from the beginning. That common thread would be an interest in sort of physical systems and how they compute and the theory of computation as it applies to physical systems. So if you look, you look outside and you see the wind blowing or you see water going down the side of a mountain or something, you can think, oh, okay, there's some dynamics to those systems. They're evolving over time. If I set them up differently, I would get a different result. So if I move the beginning of the river to a different location, it ends at a different location. The path that it follows would be different. And so you can think of these things as computations. You can think of light bouncing off mirrors and going through lenses as computing. And that's actually what I worked on in my PhD. So the theory of what you can do with optical computers. I guess, I was interested in so there's a kind of more than a sequence of institutions, there's a kind of a sequence of forms of computation that I've been interested in, and that sort of better maps out what actually happened. So I started out thinking about optical computing, then I moved on to thinking about simple universal models of computation like Turing machines, in particular small universal Turing machines, cellular automata like Rule 110 and post-tag systems. So I worked on those with my first PhD student, Turlough Neary. Then I moved on to work on things like membrane computing and Boolean circuits with Niall Murphy, another form of computing. Got into the theory of selfassembly around 2009, so that's about 10 years after I started my PhD, and got really into a topic called intrinsic universality, and then I got into molecular robotics, and then finally ending up by getting into trying to build some of these things in the lab.
So how did you get interested in the experiment, and how was the experience of doing something completely different than the theory part, and how was the transition for you?
Okay, so there's a few different answers to that. So even though I was a theoretician, I was generally okay with doing things with my hands, so I'm not the best at carpentry or metalwork or whatever, but I grew up on a farm, and so we just, did stuff with our hands. There's something wrong with something, you fix it yourself, whether it's a tractor engine or a gate or something, or a cow sick. And so I guess some theorists would not be practical minded. I guess I'm a little bit practical minded. A second thing would be, so I'm married to a biologist, and I got to see there what significantly difficult and challenging experiments looked like. And then I looked at what was going on in DNA computing or molecular programming. I was like, that looks easy. So I would have started that in Eric Winfrey's group. So I went there in 2009. And yeah, so I just thought it was possible to, like one of the first things I wanted to do was build a kind of 1D cellular automata that never really happened. But it just seemed like, hey, it's not that hard to go from a reasonable theoretical model of computation to building it with DNA. And so then I just got going at it.
So it's not that hard. Did you meet any challenges during the process?
Yeah, so I guess I said it wasn't that hard, but it takes a little while to get certain skills. I think it came naturally to me to do things like pipetting and having a well-organized system for your test tubes and so on, and trying to think of clever ways to organize materials to minimize the amount of work you have to do. Were there any particular challenges? Okay, one challenge I see. So sometimes people in our field, especially they do self-assembly. So I've done self-assembly and molecular robotics. So generally you'll use an instrument called an AFM, atomic force microscope. And some people say, oh, it's kind of tricky to use and so on. So I'm trying to, at the moment, just get rid of all that trickiness and just completely nail it down. So in my opinion, a good experimentalist should completely understand their instruments and their instrument and all its quirks and everything, and actually be in control of it and not have it in control of them. So if it's a qPCR machine, you should understand, how does the illumination work in this machine and what's the limitations of it and so on. And if it's an AFM, you should understand, well, my sample's been moved around, or else the tip is moving around. How is that happening? And can I try to fully understand things? So I think there's an important challenge there for everybody that's an experimentalist, that if you really get to know the instruments properly, just overcome that challenge, you'll get data that you can understand better, and your experiments will be more, let's say, predictable. You're reducing the amount of variance and craziness from the outputs of your experiment.
So when you say about getting to know your instruments, is that kind of, let's say, an AFM says a class of instrument, or your particular AFM? Because if I've heard right, I think AFMs, every time you change the tip, you have to break them in, and there's a lot of hassle. I guess what I'm getting at is, does this get in the way of reproducibility if you need to kind of really get to know your particular instrument before you can interpret the results?
Yeah, so, okay, first of all, I'd say reproducibility is the key. You want to sit down at an instrument, like a microscope or atomic force microscope or a light microscope or whatever, and get reproducible results. So primarily get to know your particular instrument, right? So you generally don't have access to other people's instruments, but by doing that, you'll learn more. I don't think that harms reproducibility if you're transported to another lab. So you're a PhD student in one lab and you get to know one particular instrument really well, and then you go to another lab and you see a different version of that instrument. It typically is the case that, maybe 90% of what you learned in the first lab can be immediately moved over there, and then you have to learn some final 10%. Yeah, so get to know your particular instrument in your lab. That's the primary thing.
So you've gone to one of your particular experiments with AFM. One of your well-known papers was with Dave Doty and some others on so that the name of it was Diverse and Robust Molecular Algorithms Using Reprogrammable DNA Self-Assembly.
Snappy title.
Yeah. It's received a lot of attention in the popular press. Could you tell us a little bit about the work and why you think it made such a big impression?
Okay, so one of my favorite things about that particular piece of work is that I believe it's the first reprogrammable DNA molecular computer. And what I mean is as follows. We've got a set of DNA tiles, a set of DNA molecules that sit in the fridge. And if you come up with an algorithm suitable for the model encapsulated by those tiles, then you can just implement that model that day, that algorithm that very day. And then the next day, if you get up and you've got a different algorithm in mind, you can implement that second algorithm, and so on. And in that particular paper, we implemented 21 different algorithms. So, and at some point we just had to stop. We could have kept going, we could have done hundreds or thousands of algorithms. Just like with Python, you could keep writing algorithms forever. Okay, so what exactly is it? What we have is a kind of a 6-bit circuit model. So a kind of a type of Boolean, a class of Boolean circuits that take 6 bits as input, so no more than 6 bits. So that's the limitation of our system. And what we've given is a set of DNA tiles that will implement any circuit in that 6-bit model. So you can say that those tiles are 6-bit universal for that model. We call the model iterated Boolean circuits. It kind of looks like a cellular automaton, but it's a little bit different. So the way the paper's presented is we come up with this model, we write it down, we say, any 6-bit circuit of a certain form that you can write is an instance of this model. And then we show how we compile those circuits down, right the whole way down into DNA tiles. So we drop from sort of a high circuit level of abstraction down to a kind of a square tile pictorial type thing, where you're just tiling these sort of little ribbons. And then we bring in an error correction technique that Eric Winfrey came up with many years ago. And this helps to lower the error rate in our particular experiment and makes a big difference. And then in our levels of abstraction, we drop down to a sort of a DNA tile-based motif, like a DNA strand level of abstraction, where we're reasoning about, how will the strands be laid out and how will they bind to each other? And then in particular, how will we design their DNA sequences, which was a huge part of the project and one where I really learned a lot. And then finally, we dropped to the lowest level where we actually buy the molecules. So there were 355 DNA strands we bought and kept in the fridge. And then we had a really fun part of the project where we spent a number of months just coming up with new interesting programs and then boom, implementing them in the lab. Like, for example, just because I mentioned earlier, like when I was younger, so just like coming out of my PhD, I was working with a PhD student, Turlough Neary. We worked on something called Rule 110, which is a cellular automaton. We were able to implement a 3-bit instance of Rule 110 with these DNA tiles. So I really like that. So in the, around 2018-19, we're connecting back to something I worked on in 2006. We were able to implement, so we implemented a sorting algorithm which sorts bits. So it puts all the zeros to one side and all the ones to the other. An algorithm that told you whether a 6-bit number was a multiple of 3. An algorithm that checked for palindromes. Algorithms that copied bits and drew patterns, randomized algorithms. So wide variety of different things. So that was fun.
Do you think it was kind of the variety and the very visual nature of it? Because you have some beautiful images in that paper of kind of these long ribbons where you can see the computation happening. Do you think that's why it made such an impression in the media?
So it made an impression in the media. Yeah, I don't... I like the images. So I took a lot of those AFM images. I worked real hard to get them looking good. Yeah, I don't know. I'm just so intimately tied to the project. And I should mention that also my collaborators, especially all the people on that paper, but especially Dave Doty and Eric Winfrey, we put in it was real like Three Musketeers kind of thing. Enjoyed working with the people on the project, but I suppose it is kind of visually striking to see a tape recording of your computation. Like I'll have to say that early in the part of the project, so towards the end of the project when we actually got it working, I remember I did our first randomized algorithm, right? So we had done an algorithm that sorted bits, which I mentioned before, but basically that means you get an input that's 6 bits. Zeros and ones, and then you're making a little ribbon, and the ribbon is running so it's hard to draw pictures on a podcast. But it's like a scarf sitting in front of you, right? Lay the scarf on the table, and the scarf has been grown from left to right. And starting at the left, you've got some bits written there, and the bits are in columns. And then over time, as the scarf grows, the zeros go to the bottom and the ones go to the top. Okay, so you're sorting the bits, you're pushing the ones to the top. And then we did another one that just copied bits. So that was a really boring circuit, one of the most boring circuits I know. And all it does is just take some input bits and just copies them straight to the right. So it's just making stripes on a scarf, which is how most scarves actually look. They're either plain colored or they've got stripes, right? But if you mix those two, if you mix the tiles for those two, what you get is a circuit that both copies and sorts at the same time. And the way we set it up, it's a randomized circuit. So at each step, the system chooses, hey, will I sort these bits or will I copy these bits? So you get this mixture of sorting and copying, and it happens at a very local level. Like for each pair of bits, the system is flipping a coin essentially to say, hey, will I sort the bits? Will I move the one up? Or will I just copy the one to the right? Okay. But so over the years with algorithmic self-assembly, which is something that Eric Winfrey and his lab have worked on a lot. So in particular, Rebecca Schulman, who's been on this podcast series, has done a lot of work on that. Also, Paul Rodemund, Constantine Evans, who's now with us here in Ireland, and various other people have done amazing work on algorithmic self-assembly. And so the first two algorithms we did, I guess they kind of were, oh, right, okay, so people have done this kind of algorithmic self-assembly before. But then I had this randomized one, okay? And so I was sitting at the AFM, and the images were coming in of this randomized computation of a mixture of sorting and copying. And so the bits very slowly get sorted to the top of the scarf. And what I had there was, in front of me, a nanoscale tape recording of a large number of randomized events that were happening too quickly, and they're too small for us to see live in real time by any method that I know. But yet I have a recording of exactly what happened there. And there was thousands of these things, right? So as long as I kept AFMing that day, I would see more and more and more of them. So that was a very visually striking moment for me in the project. So it was a bit difficult to answer your question at first because I've been so immersed in it. But if I do remember back, the way they look can be very striking.
You said it takes 300 or so individual strands, unique strands, to build the 6-bit computer. How does that number of strands grow as you want to increase the number of bits?
Well, so the number of tile types per bit is 64, meaning that for each bit that you add, you'll need 64 more tile types. So scale for n bits is something like 64 times n. There's a minor correction to do with how we handle the border, so just an additive term. So it scales linearly in the number of bits. So it's cheap from the point of view of scalability. It's not like it scales polynomial in the number of bits or worse, exponential in the number of bits. It's just linear.
And so was 6 bits like just a choice which gave the best compromise of interesting algorithms and like just feasibility in the lab?
Okay, good question. We would have liked 8 bits because the power of 2, and computer scientists like power of 2. Okay, so what the history of why it's 6 bits comes down to actually a sort of technological limitation that we bumped into at the time. So we can fit 6 bits onto, so our scarf that I was describing, if I want to be fully truthful, it's not growing on a scarf, it's actually growing on a tube. And so the top is bound to the bottom of the scarf, so you get this long tube. So we're growing a tube from left to right, and along that tube, or that cylinder, the computation is happening as the thing grows. This tube is made of DNA. So running along the long axis of the tube, you've got these DNA helices, and there are exactly 16 of them. So basically, we managed to make 16 helix nanotubes, and we did try for a little while. There was a student who worked with Dave on trying to scale up to 32 helix nanotubes. That kind of didn't work out at the time. Now we've learned a lot, and I think we could go back and hit that problem again, and we might know a bit better about what to do. But at the time, 16 helix were one of the largest nanotubes we had made, so we just ran with that. A second thing that's kind of important is to mention another student, Joy Hoy, who was working with me on my first experimental project, which is still not published. And we were working with nanotubes there trying to make a self-replicator. We needed to characterize the growth of these nanotubes in a special way. In particular, we wanted to characterize the nucleation properties of these nanotubes. So basically, at what temperature do the tiles start to come together to make these DNA nanotubes, and then also to give a measurement of the kinetic barrier to nucleation for these nanotubes, because we wanted to do something called seeded growth, so have a seed that grows a nanotube off it. And at the time, I think 16 helix were the largest nanotubes that Joy and I had made, and they were made using a motif from a paper by Pong Yin, Rizal Hariardi, and many others, called the single-stranded tile motif. And so, 16 helix were the largest nanotubes working for us at the time on a different project, and then we switched over to using these for algorithmic self-assembly.
I heard that there's some constraint about because the tile grows in the tube, so there's a limit on the number of bits that you can use. Is that right?
Yeah, that's right. So there is, and one thing one might ask is why a tube, and why not go for something flat and try and get rid of that size limitation? And that's actually something we're trying to do now. So tubes have been great for DNA self-assembly. There's something about them. They're just wonderful. We really like them. And I've worked with tubes a bit. Rebecca Schulman's lab has worked with tubes a lot. A bunch of other people have worked with tubes. Anyway, so we'd like to get better at moving away from tubes. Here's one thing that's really nice, and here's why we used tubes. Okay, when you're growing a DNA nanotube, the growth front, or the growth frontier, it looks, the quick way to say this is it looks the same everywhere. So basically, if you start putting tiles in there, there's no special border anywhere. So there's no top or bottom, or there's no border. But as soon as you start doing flat things, like if you think you've got let's say you've got a DNA origami, right? Everyone in the field knows what a DNA origami is. There's something different about the staples along the edges compared to the staples on the inside. And so likewise with tiles, if you're building a DNA tile nanostructure, the tiles on the outside, they might be a little bit different. And if you're really trying to control the growth order of things, which you want to do with algorithmic self-assembly, then that kind of stuff really matters. You're going to have it be the case that the energetics of binding for tiles on the edge of your structure is exactly like the energetics of tiles binding on the inside of the structure. And that can be very hard to achieve, and or take a lot of maybe try a bunch of design iterations to eventually achieve that. So it's sort of a non-trivial problem to solve. There have been some algorithmic-based systems with a border on them, and that are actual ribbons, like ribbons made by Constantine Evans in his PhD thesis, for example. And so they've got a special double tile motif that's used there. But doing that more generally to make structures that have arbitrary flat 2D shapes is sort of an open problem for algorithmic self-assembly.
So this was quite an international collaboration, and you've actually, as we mentioned earlier, you've been to a lot of different places and countries, France, the US, Ireland, Spain. Have you noticed any big research culture differences between all of these different places?
Okay, there are massive differences in terms of the kinds of positions available as a researcher. So these are not research differences. So if I take the word researcher and I abuse it now and make it the word, let's say, academic, then there are differences in how the kind of positions available and how you go about getting positions, in particular permanent positions. In terms of research culture, yeah, there can be differences. There's differences amongst institutions, and there can be a little bit of variability by country. But generally, scientists around the world that I've interacted with, we all have the basic sort of aims and goals. We go about problems in very different ways, but we all want to do good science, but it can be packaged a little bit differently in different countries.
And with respect to permanent positions, you started your group at Inria in France. What was it like moving from France and bringing your group with it? Did that have any unique challenges?
Okay, so moving countries in general, I'm okay with that. So I've worked in a variety of countries, and I've really enjoyed working in those countries, mainly because you get to eat their food, and places like France and Spain and Southern California all have great food. So that's really good. In terms of actually, let's say, moving the group from Inria to Ireland, I don't think it was particularly challenging at all, because we had a shared lab space we were using while I was in France. And it wasn't actually in Inria, it was in another institution with André Estevez Torres and Jean-Christophe Gallet, who were kindly, their groups are in Paris, and they had given us some space to use. And Inria itself is an amazing place, but it's computer science only, essentially. So it doesn't have wet labs, for example. And so what we were moving in terms of wet lab infrastructure was not very much at all. It was sad to leave France, and I was sad to leave Paris and my colleagues there, but we still keep in touch somewhat. We, in particular, Nicolas Chabanel, who's now at Lyon, has a group, and we have regular group meetings together, actually. So that's one really good thing that came out of that. There were two people who came with me, Pierre-Etienne Mounier and Tristan Sterren. So Tristan is a PhD student, still with me here. And so it was probably challenging for them to move country, and that's a great debt we incur to people when this happens. I think the challenge then was setting up a lab in Ireland. So the way it worked for me, and I think when it comes to getting academic jobs, there's different ways you do it in different countries and in different parts of the world. I think for Europe specifically, generally you don't get a startup. If you get a permanent academic position, you don't get a startup package the way you do in the US. But the funding is a little different. There are opportunities here in Europe. So for me, I managed to get an ERC award, which was significant enough to fund a group for 5 years and pay for an atomic force microscope. And then in coming to Ireland, the Irish government gave me additional assistance, which was rather significant. So the organisation is called Science Foundation Ireland. It's sort of like the NSF in the US, but for Ireland. And so they give funding to kit out our lab and to build our lab. So the main challenge wasn't the move itself. The main challenge was over the next year or two building out the lab. But that's also a very fun challenge, because if you do experiments, you often have particular instruments you want to get. And once you've got the funding, you can get those instruments.
I guess bringing your group members along is kind of an upgrade from the two-body problem to an n-body problem in academia.
Yeah, that's right. Like when I moved to Spain, I was working with Turlough Neary, who was by then probably a postdoc, I think, and he had a position somewhere else. We were kind of working remotely, but I was working very closely with Niall Murphy, who was a PhD student of mine, and Niall decided to come to Spain, and I think Niall was the kind of person who liked traveling anyway, like myself. But then actually he met his future wife there, so I think that worked out great, actually.
I wonder how did you feel about traveling a lot and not in a state that is not so settled before finding a faculty position? Is it because you just like traveling a lot, so it's not a problem for you?
Yeah, I think so. Yeah, I do like traveling a lot, so it's not a problem for me. That is true. It was never really a problem. It was always like an opportunity, something I really enjoyed doing. But I have a wife, and we would have repeated two-body problems to solve, which we generally did solve. So, for example, we both went to California. We both got positions at Caltech. That kind of thing can be tricky to solve in general. But anyway, yeah, I've really enjoyed going and working in different places. And so, it means also when I go back to those places, they've kind of a deeper meaning for me, I guess, So I know when I go back to Seville in Seville, I know what kind of food I want to go and try out.
Since moving to Ireland, what are you most excited for your group for the future?
I think mostly helping the people in the group realize their projects. The projects we're undertaking here, they're kind of non-trivial. I guess it's the same everywhere, but we try to do meaningful projects that we think, life is short, so you got to pick carefully what you work on. So helping people to realize those challenging projects, that's one thing I'm really looking forward to. So seeing those get finished, it's always hard to finish off major projects. I have examples of unfinished projects. I tend to gravitate towards projects that they're so big, sometimes they're personality changing. You come out of it and you're a different person than when you went in, and you've really learned a lot. And sometimes it's a different part of science that you've worked in, or sometimes it's just a new part of mathematics that you needed to learn or work with. So yeah, I'm looking forward to doing lots of that. I'm
kind of curious, following up on what you said, what makes it hard to finish a project?
Oh, wow. Okay. I could probably ask you the same thing, Boya or Georgios or Will. I bet you everybody has examples of unfinished projects here. Okay, I'll try to be concrete. So in 2009, I took a position as a molecular programming fellow, I think was the name of the position. It was a postdoc in mainly in Eric Winfrey's group, but also in Shuki Brooks' group in Caltech, both of which are wonderful people and great scientists. And I started working with Eric and somebody called Bernie Yerke, who's rather famous in our field, as he was one of the inventors of toe-hold mediated strand displacement, and brought that to as a technique which later was used to build strand displacement circuits, for example, and many other things. And we tried to build a self-replicator. We made some really great progress, but we never quite published it. We never quite finished it off. And so why didn't that happen is a kind of a rewording of your question for that particular project. The data wasn't quite there. It was actually my first experimental project using DNA, and it was a little bit stochastic and noisy. It was unlike many other projects we have. It involved use of duct tape and building our own apparatus in a glove box, drilling holes in that glove box, having pipes go in. We were bubbling in a solution, controlling the temperature in this giant glove box and the humidity and all this crazy stuff. So for my first project, we kind of built the setup together, and the results were mixed. So sometimes the experiment would work beautifully, and then sometimes it wouldn't work. Something would go wrong, and we tried to figure out, what's going on? And we realized, this home-built setup we've built, there's some imperfections here and there, and one of our enemies in this project was spurious nucleation, and that would happen sometimes. So I really wanted to get that project in a setup so that it worked in a much easier way, and it worked 100% of the time. And because of that, it never really got finished. And then we were moving along, and then we had this opportunity to do this algorithmic self-assembly project with Dave Doty, who I mentioned earlier, and with Eric and some others. And I kind of switched over to that and left that project unfinished. But we're going to continue it now, but I think in a better way. So we've had time to step back, and we've completely rethought the project. We've got new ideas about how to do it in a much better way. In particular, there's a postdoc here, Trent Rogers, who's had some great ideas on how to better control, let's say, nucleation, which is a fundamental problem in DNA self-assembly anyway. And another postdoc recently started with us, Ismail Muller-Ruz, has started working on it actually. So I'm ready to see the version 2.0 of that take off. Sometimes projects don't get finished because they're boring. So that doesn't happen to me so often, but I think I've got about 10 or 20 folders there of my computer of different things that didn't get finished. And yeah, maybe it was something in theory and it didn't turn out to be that interesting. So you ask some question and then the answer is easy. And you're like, okay. So I'm not sure I want to follow up on that. So there's also that as well. So it's very different for theory and experiment, I think.
When research occasionally gets to the point that it's very boring or you need a break, is there anything you do kind of outside of academia and work?
Okay, so research never gets boring for me, actually. I guess I was referring there to particular projects where it quickly went to something not interesting. So then you just stop thinking about that and you think about something else. What else do I do besides research? Well, I have a job here where here's some non-research things I do. Research administration and research and accounts and hiring of people and all that kind of stuff. But that's really not what you're asking about. I play a bit of drums. Might try to record an album with some old friends of mine. We tried to be rock stars when we were teenagers, and that was that kind of didn't work out. I became a scientist instead. So I'll play some drums. I like to go cycling, but I just kind of always cycled a little bit. Like I used to cycle to school when I was a kid, but now I actually get on a bike, and I went 100 kilometers the other day, so that was fun. But yeah, just chilling out, hanging out with my family. There's been a lot of that since coming back to Ireland. There's been a lot of that.
Do you think teenage Damien would be disappointed that you're a scientist rather than a rock star?
Yeah, good question. Yeah, that's a really good question. I don't know. I'd like to think not, but maybe he was a bit of an idiot. So.
At least maybe when you get your album out, you can call yourself one of the few true rock star scientists.
Yeah, but are there any rock star scientists who've actually become rock stars and aren't just scientists who sometimes play music?
I think there is. I think the guitarist from Queen has a PhD in astrophysics.
Yeah, okay, yeah, that's right. And actually, Bad Religion, singer of Bad Religion, scientist. Christos Papadimitriou is a famous theoretical computer scientist who has a band. Never seen him, but yeah. So yeah, there are some musician scientists out there.
How do you cultivate a multidisciplinary environment in your research group?
Yeah, so what you can try to do, so it's different for different groups, I guess. One can try to hire people from a variety of backgrounds. So when we advertise positions, we specifically say that we take from a wide variety of backgrounds. So that's one thing. Second thing is maybe if somebody has a strength in theory, you might try and encourage them to look at papers that are far from their interests, but yet might sort of gel with them somehow. So that's something else you can do. I think having a vibrant group meeting is important, and having a vibrant and coherent group meeting, because ultimately what will help people both like in presenting their own work. So it helps you if you can talk about your own work and get feedback on that from people who have a variety of backgrounds. And then it works the other way when you see those other people talking and thinking, and you see the kind of questions they ask that might help inform you about, like if you're mostly from an, let's say, biology background or physics background or chemistry background, and you don't know anything about computer science, and then you see computer scientists ask certain questions in certain ways, and you see what they like, it might help you think in that way. I think another thing is to encourage folks from a theory background to try an experimental project and vice versa, and in particular to pick something that's like going to be a paper, so then you're really well motivated to get moving at it.
So Damien, what would be one of your favorite things to implement?
Okay, so in the lab, first of all, I'd like to implement the things we're trying to implement right now, which are, I'll get to the answer in a second, but I should say that we're trying to build reconfigurable robotic systems. So Kai Wood, a PhD student here, is working on that. He's working on implementing a model called Turing machines, which are like Turing machines, except they've got an N in there, and they're actually a robotic model. I mentioned implementing self-replicators, and Ismail and Trent are working on that. And then more precisely controlling the dynamics of self-assembly, and maybe even building fancy algorithmic self-assembly systems. Trent Rogers, Constantine Evans are working on that. And then cool ways of computing, new ways of computing. Tristan Sterrin here and Abir Eshra, a postdoc, are working on that. But besides all those things, in the more distant future, one thing I'd like to implement would be an intrinsically universal tile set. And so this ties back to some of my favorite theory work that I've worked on. And so an intrinsically universal tile set is a tile set that's able to simulate a wide class of other tile sets or other tile systems. Okay, so we know you can have so we know that the operating system on your computer, it could run other programs. It can even run other operating systems, like in a virtual machine or whatever. And we know from theory that, so Alan Turing gave us the Turing machine, what we now call the Turing machine, but also showed that there are universal Turing machines. So you could have a universal Turing machine that simulates any other. We know that tiles, algorithmic tiles, so early results by Eric Winfrey and related results by Paul Rodeman and David Soloveichik showed that you could use tiles to run Turing machine programs and in fact use that to build shapes. So you could encode a shape inside an algorithm, a Turing machine, and then have some tiles come together and run that Turing machine to build a shape. So that's pretty cool. It's a kind of universality, right? You're simulating Turing machines there. But what happens, like imagine now you want to have a kind of self-universality where you've got a set of tiles. And what that tile set can do is if Boya has a tile set, and let's suppose it builds a tree, starting with the roots, it builds a trunk, and then it builds the branches and finally the leaves. It's making a 2D picture of a tree, let's say. And Georgios has a tile set that builds a smiley face, starting at the top, the forehead, and going all the way down to the chin. And then Will has a tile set that builds a house, say a picture of a house. Well, I want to have, what I could have is a tile set that could build any one of those things depending on how I program it. And by program it, I mean I pick out some of the tiles and I lay them together to make a seed. And then from there, it just grows to make one of those three things depending on how I set that seed exactly. And so we wondered if there was such a tile set, theoretically, and this is how I got into self-assembly, actually. So one of my favorite things to do, or one of the most important things in science, is coming up with good questions. And this was one of my favorite questions. And the question we had was: is a particular tile assembly model, is it intrinsically universal? Does it have one of these tile sets that could build anything you want, and not only build the shapes but also do the dynamics just right? So I said Boya's tiles built the tree from the roots up to the leaves. So I want a program system so it will build the tree from the roots to the leaves and then be able to reprogram it so it builds the tree from the leaves down to the roots. And so you can precisely not only make shapes but also capture the dynamics of any tile set that you're simulating. So I'd like to implement an intrinsically universal tile set. The problem is we worked for quite some years, and I say we, it was myself, and then I met a bunch of people from Iowa State University, which was Jack Lutz and his 3 PhD students, Matt Padditz, Scott Summers, Dave Doty, and then later Robbie Schweller. We all worked together on this project, and we found, after many years of working and trying to prove whether or not there was such a tile set, we came up with the answer: yes, there is such a tile set. So we found a tile set that is intrinsically universal for the abstract tile assembly model. So it's a theoretical result. But by our construction, the tile set would have many millions of tiles. And so earlier we talked about the fact that I had myself and my colleagues had implemented a system that was 6-bit universal, but had 355 tiles. So now if I want to implement a tile set, the only known tile set intrinsically universal for that model, the abstract tile assembly model, that would require millions of tiles. It could be tens or hundreds of millions, by the way. So we didn't actually compute the exact number of tiles because the paper is at a higher level of abstraction than that. But we did show that such a tile set exists. But I could imagine a project where one scales that problem down. Like I know that Matt Patta's group later on in the University of Arkansas figured out that, hey, there's a system with about 100,000 tile types that's intrinsically universal for... there's a tile set with 100,000 tile types that's intrinsically universal for the 3D version of the ATAM, the abstract tile assembly model. So 100,000, okay. If we could scale down to, let's say, 1,000 and find some sub-model that is able to do interesting things. It can, I don't know, simulate Turing machines, make a variety of interesting shapes, but not the full expressivity of the tile assembly model. Then, to build that would be something else, because now instead of having 355 tiles to implement any 6-bit circuit, I would have 1,000 tiles that can implement a huge variety of tile assembly systems. So I wouldn't need to design individual tile sets to do different things. I would just have this one tile set in the fridge, and that could keep us busy for many years to come.
Sorry. Are those people saying that it's they've shown that the smallest number of tiles for a universal tile set in 3D is on the order of 100,000, or could there be
there could be a much smaller. There could be a smaller
okay.
one. They're actually showing the opposite. What they're showing is, with 100,000, it's enough to do the job. So maybe you could do it for the 3D model with 10,000 or less. So generally, the results so the theoretical results on when you give a positive construction in tile assembly, you'll give a construction, and then maybe later you could improve it and reduce the number of tile types. In fact, myself and Tristan Sterren and Matthew Cook just had a paper at the recent DNA conference where we used 4 tile types to simulate any Boolean circuit, but we kind of cheat by allowing the seed to be a disconnected structure, meaning that you can imagine you've got an origami with little blocks that are on it already, and then your tiles are going to attach to that origami, attach to that surface, and flow through, like flowing through a maze. Imagine someone has built an arbitrary maze, but they've put little annotations on the wall that says, oh, if you want to walk through this maze, you got to solve some Boolean gates. And by doing that, you could use a maze to encode a circuit, and then you can have tiles flow through the maze, running the circuit that's encoded by the walls of the maze. And it turns out you can do that with just 4 tile types. So depending on the setting, you can get really small tile sets to do interesting stuff.
But do you conjecture that there is an experimentally feasible universal tile set?
Yeah, except... So we tried it. So actually, I had a summer student here, Dara McConville, come work for a summer and try to find, what could you do with like 100 tiles or something like that, And it's not a straightforward problem. So I think it's a problem that has legs theoretically. So like, as in, we have to sit down and do some theory to try and find something on the order of 1,000 and characterize what it does. But yeah, I would guess, I would say that, it is possible with 1,000 tiles. You can really do a lot of stuff. And then conjecture is the wrong word here, but I also would guess that we could implement something with that number of tiles in the lab in the next, not today, not tomorrow, but in the sort of next 5-10 years sort of scale.
This reminds me of the history of aperiodic tilings, where they started with finding, say, a set of 20,000 tiles, and then by the end, I think it was Roger Penrose, did he come up with it in 2 tiles? I'm wondering if you have any intuition of what the lower bound on, or what the optimum number of tiles for intrinsic universality might be.
Okay, so the answer is 1. Okay, but here's why. I'm going to cheat, all right? If you allow my tiles to be not square, all right, which is reasonable here, we're talking about very small numbers of tiles, all right? Then I can give you 1 tile that simulates any Turing machine or any tile assembly system. Okay, right. I'm going to have to say a little bit about that, right? Because it's one of my favorite papers. By the way, it was rejected the first few times we tried to submit it, and some of the rejections seemed to be, it looks like the authors here are having too much fun. This doesn't seem serious to me. So the paper is called One Tile to Rule Them All. And basically, we have a tile that has many sides on it, right? It's a regular polygon, but it's got millions of sides, all right? So a square has 4 sides, a hexagon has 6, ours has hundreds of millions, okay? And again, we didn't try to compute the exact number of tiles, but what it does is it simulates the intrinsically universal tile set I mentioned earlier. And what happens is, if you pick up this tile, it looks almost like it's colored golden, by the way. So it looks almost like a golden ring, right? So that's why it's one tile to rule them all. And so let's say it's got n sides. Then it's got n rotations, right? It looks different on each of the n rotations. And each one of those rotations is kind of going to act like a bunch of square tiles together. That's sort of the way to think about it. In fact, each one of those rotations acts like a different square tile. So we showed with those tiles that are able to rotate and have many sides, just one of them is enough to simulate any tile set in the Tilesembly model. But just coming to what you said, Will, about aperiodic tilings, right? There's also an interesting connection there, because in the end of the paper, we made this connection to aperiodic tilings, and we showed that one tile, when it has something like, I'm going to get this wrong, but 44 sides, something like that. There's a single tile that has aperiodic tilings with a small cheat that we need to allow holes in the tilings. So we've got this regular 40 gon or 44 gon, and each different rotation simulates a square Wang tile, and you take a small aperiodic Wang tile set and you can just simulate it with our system. The fact that it cheats by putting small holes in there is not a huge problem in my view, because there are other ways to get aperiodic tilings with one tile. So Penrose has I think they're called the kite and the dart, the 2 tiles. There is the Sockler-Taylor tile that kind of looks like a hexagon, except it comes in a few flavors. So you've got this single hexagon, but it's actually so there's a 3D version. So this one cheats by allowing for 3D, and then you look at its 2D tilings and they're aperiodic, which means that they don't repeat in a certain sense. So if I'm standing on the tiling and I look out to infinity, I see a certain pattern, and then if I move to another part, I take a step and move to a different part of the tiling, I see a different pattern, right? So that's what aperiodic means here. So the Sockler-Taylor tile is a single tile that looks kind of like a hexagon, but it's a 3D thing with some extra gadgets attached. And when you tile with that thing, you've got one tile that can tile a plane, but it's cheating because it's 3D. You can have a disconnected version of the Sockler-Taylor tile in 2D, which is kind of just like the 3D one. It also is aperiodic. And then there's 1 or 2 other single tiles that cheat, and one of them is ours. So the way we cheat is by allowing holes between the tiles. So I thought that was kind of cool that we did some self-assembly stuff, and then we were able to link it to more classical tiling theory. So that was fun. That was with Eric Damien, Martin Damien, Chandra Friquet, Matt Padditz, Robbie Schweller, and Andrew Winslow.
Is there any chance of implementing tiles with holes in them in DNA and 40 sides?
Okay. Yeah. So holes are not a problem. 40 sides is a big problem. So basically, if you think about, if you're going to use DNA hybridization, right, you want to be binding by at least, let's say, 5 bases. And in fact, you'll need more than that to get some orthogonality. So then suddenly the thing that you're talking about has scaled up a lot. And so then I don't think it's a super good direction for going with many sides. In fact, one of my experimentalist friend used to we'd go for lunch together, and he'd just really enjoy laughing at our tile that had hundreds of millions of sides. But, it's a beautiful, beautiful theoretical object. And that's like an important point. I think that some experimental, when it comes to theory and experiment, I know that some experimentalists feel that the theory should somehow be in service of experiment, or meaning that do theory because it'll help us understand what is possible with experiment or not. And I definitely agree with that. I sometimes use theory in that way. But I also really, really enjoy theory just for the beauty of it. When you find kind of something unexpected, like a proof that's really nice or that was very challenging to get to and then really surprised you with the way it worked out. I think that's one kind of scientific inquiry. That's one mode of scientific inquiry that's very satisfactory for me.
Thank you so much for joining us, Damien. Stay tuned to our newsletter for details on our next podcast episodes. Thanks for listening.